So in the previous page, we discussed how the aldosterone increases the sodium retention by this genomic action. Now, there is other function of the aldosterone that is by the non-genomic action of the aldosterone, which is that the aldosterone also binds to the cell membrane, and by a rapid non-genomic action, it increases the activity of the sodium-potassium exchangers. What is that? Sodium-potassium ATPase, you know already. So thus, it increases the intracellular sodium ion concentration, and the second messenger involved in this process is inositol triphosphate, or in simply IP3. Now, in any case, the principal effect of the aldosterone or the sodium transport takes 10 to 30 minutes to develop, and it take peak even later. So it is suggestive of the synthesis of the new proteins by a genomic mechanism. Now, the relation of the mineralocorticoid to the glucocorticoid receptors. It is a very interesting thing. Now, in vitro, that means in case of research, when we take some samples and test outside of the body, the mineralocorticoid receptors, or the MCD-R, has an appreciably higher affinity for the glucocorticoids. Remember, maybe they are the mineralocorticoid receptors, but they have much more of an affinity to the glucocorticoids rather than the mineralocorticoids. So, even the glucocorticoids are present in large amount in vivo. In the body, inside the body, the glucocorticoid concentration is much more than the mineralocorticoid. Now, this raises a question why the glucocorticoids don't bind to the mineralocorticoid receptors in the kidney and the other locations and produce a mineralocorticoid effect. Now, the answer of this question is that the kidneys and the other mineralocorticoid sensitive tissues also contains the enzyme 11-beta hydroxysteroid dehydrogenase type 2. Now, remember, in my previous note, it was seen that the 11-beta HSD2 is responsible for the conversion of the cortisol to the cortisone, but not vice versa. Remember, cortisol to cortisone production is done by 11-beta HSD type 1 and also type 2, but the reverse production of the cortisone to cortisol is already or only done by the 11-beta HSD type 1. So, in case of the 11-beta HSD type 2 present in the kidney and other mineralocorticoid sensitive tissues, this leaves the aldosterone untouched because it converts the cortisol to the cortisone, as you already know, and also the corticosterone to its 11-oxy derivatives. And these two products, that means the cortisone and the 11-oxy derivatives of the corticosterone, never bind to the mineralocorticoid receptor. So only what is left to bind the mineralocorticoid receptor is aldosterone. So glucocorticoids, even they have the much more affinity to the mineralocorticoid receptors, never bind to the mineralocorticoid receptors due to the presence of these enzymes only. Now, we will discuss a very interesting thing here, is the apparent mineralocorticoid excess, or in short form AME. Now, if the 11-beta HSD type 2 enzyme is deficient or it is totally absent, so there will be the cortisol has a marked mineralocorticoid effect. Now, easily you can now guess that the cortisol will never be converted to the cortisone, and as the previous page already told you, the cortisol is converted with the help of this enzyme to the cortisone, and it never binds to the mineralocorticoid receptors. So in the absence of this enzyme, the cortisol have much more mineralocorticoid effects, and the resulting syndrome is known as this apparent mineralocorticoid excess, or in case AME. So what will be the clinical feature? It will be the hyperaldosteronism. Like the hyperaldosteronism means the aldosterone level is high, but here the aldosterone level is not high. Here the glucocorticoids are doing the effects of the mineralocorticoids. So it means that mineralocorticoids is not excess, but the clinical feature is like the excess mineralocorticoid. So there will be the feature is like the hyperaldosteronism, as cortisol is acting on the mineralocorticoid receptors and the plasma aldosterone level as well as the plasma renin activity is low. Because there is no increased production of the aldosterone, there will be no increased production of the renin to convert it to back to the normal situation. So AME can be due to the congenital absence of the 11 beta HSD type 2 enzyme. So what is the treatment? Remember, the prolonged ingestion of the licorice. Licorice means joshti moto, jeta amra onek shomoy খেয়ে থাকি নানা রকম রান্নায় বা সর্দি কাশিতে, can also cause an increased BP. And outside the USA, many countries contains the glyceretinic acid inside this licorice, which inhibits the 11 beta HSD type 2. So person taking this type of licorice for a long time have increased mineralocorticoid receptors, activated sodium reabsorption via this ENAC in the collecting ducts and thereby increasing the blood pressure. So very interesting thing, AME or apparent mineralocorticoid excess. Now, what are the different effects of this adrenalectomy? Adrenalectomy means if we remove the adrenal gland, what will be the different changes on the body or body metabolism or different type of electrolytes. In adrenal insufficiency, that means if there is no adrenal gland in our body or deficit hormone production by the adrenal cortex, there is sodium loss in the urine. As we know, the aldosterone is responsible for the sodium retention. There is no aldosterone, no sodium retention. Sodium is lost in the urine and potassium is retained. So there will definitely increase plasma potassium ion concentration, and when this adrenal insufficiency develops rapidly, the amount of the sodium lost from the extracellular fluid volume is much more than the sodium excretion in the urine, and this is suggestive of the sodium is also entering into the cells. So when the posterior pituitary is also intact in case of the adrenal insufficiency, the salt loss is much more than the water loss, because we know already the posterior pituitary has a hormone secreted, namely ADH, antidiuretic hormone. So due to the presence of the antidiuretic hormone, water loss is very less than the salt loss. So therefore there will be the plasma sodium ion concentration going lower down, and however we know the plasma sodium ion concentration is the only thing which retains water as well. Sodium attracts water to retain inside the extracellular fluid compartment. So whenever the sodium concentration inside the plasma is going down, the plasma ECF volume is also decreasing, and thereby the features are hypotension, circulatory insufficiency, and thereby producing the shock, circulatory shock. Now the changes this due to the adrenal insufficiency can be reversed by increasing the dietary intake of the sodium chloride. Now the rats can indefinitely live on the extra salt diet if the adrenal insufficiency occurring in them, but remember the dogs or the humans can survive on this extra salt diet alone until and otherwise the mineralocorticoid treatment is being started for them. They will die due to the absence of this mineralocorticoid and loss of the sodium from the extracellular fluid volume, and they will collapse. Now it's a very important short notes which I am going to tell you is the secondary effects of the excess mineralocorticoids, or this question may come as a escape phenomena. The prominent feature of the prolonged mineralocorticoid excess is potassium depletion due to the prolonged potassium diuresis. We already know if there is mineralocorticoid excess, there will be much more sodium retention in exchange of the potassium. So potassium is going throughout the urine and there will be potassium diuresis, potassium loss. H plus ion concentration is also lost in the urine. To remember to save or reabsorb the sodium, kidney also excretes the potassium ions and H plus ions in exchange of the sodium. So sodium is retained initially, but plasma sodium ion is increased slightly at all due to water is retained with osmotically active sodium ions. That means whenever there is sodium, there is water molecules, as we already discussed. So therefore, the increased blood pressure, increased extracellular fluid volume occurs. Now it is seen as a very interesting fact that when ECF volume expansion passes a certain point, sodium excretion is usually increased. Even there is increased mineralocorticoid. We know mineralocorticoid is there, sodium should be retained, but after a critical point, the sodium excretion is occurring rather than sodium retention. So this is called escape phenomena. It's a very important short note. Increased secretion, and why this is occurring? This is occurring due to increased secretion of the ANP, atrial natriuretic peptide, which is secreted from the atria of the heart. Thus, this actually the function of the ANP to increase sodium excretion when there is increased ECF volume, and mineralocorticoid don't produce the edema. Even there is in the normal person there is increased mineralocorticoid, and even in the persons who have the hyperaldosteronism. So due to the escape phenomena, even there is a mineralocorticoid excess, the sodium lost in the urine, and thus it prevents the formation of the edema. And this escape phenomena, this doesn't occur in some disease states. In these conditions continued the increased ECF volume expansion. In some cases, it is seen that the ECF volume expansion still occurs even there is protection barrier by this ANP. Even in certain diseases, this ANP protection barrier of escape phenomena doesn't work, and there is a formation of the edema.